Camera's off again. So you're going to now continue looking at the segmentation gene. And the example we are going to look at now regarding segmentation gene are Wingless and Hedgehog. An interplay between Wingless, Hedgehog, and engrailed, it leads to a beautiful pattern which highlights the segmentation in growing larvae as well. We call them denticle belts. Denticle belts on ventral side of cuticle. Okay. And as I said, so each segment in larva, it has very well-defined anterior and posterior pattern. And that pattern is based on this denticles. Now, each segment, so this is one segment from here till here. The interior part contains the denticle belt, while the posterior part doesn't. Another very promising thing is that the pattern of each denticle belt is unique. I mean, these are basically projections from cuticle like this. And when we will zoom in, we will see that the denticle belt of this segment is unique and does not match this one, and so on. If we have mutation in wingless is a segmentation gene. So we will see denticle belt also here. Normally, which is naked, which is without any denticle belt. What we see that if we have mutation in wingless, we'll see a mirror image of this denticle belt here as well. So the compartmental boundary in a segment is disturbed, and cells which have attained different fate here, they actually become similar to this one in the absence of wingless. And if you have hedgehog, you have the same phenotype as well. Whether you have delta wingless or delta hedgehog, phenotype is same. It means they are related. At molecular level, what they have found that, I mean, this is just a wingless and hedgehog, just a repetition. So wingless ligand, when it binds to the receptor, the signal transduction pathway through disheveled transduces the signal, which leads to release of beta-catenin transcription factor. Beta-catenin moves into the nucleus and binds to wingless responsive genes. In the absence of wingless, beta-catenin is degraded and does not move into the nucleus. The hedgehog, we talked about that the past is the receptor for hedgehog. So when the past binds, smoothened is not able to hold hedgehog signaling anymore. Now, as a result of the signal transduction pathway, cubitus interruptus is released. It moves into the nucleus and activates DPP, wingless, engrailed, etc. genes, which are hedgehog responsive genes. At molecular level, what we have found, how the interplay between wingless, hedgehog, and engrailed play a role in the denticle pattern formation is that, so if this one is the parasegment boundaries. So this is one parasegment. This is next parasegment. This is next. These are independent parasegments. Okay. Now, if we consider this side as the anterior of parasegment and this is posterior of parasegment. So now this posterior of this parasegment and anterior of next parasegment, this is going to make one segment, adult segment from here to here. The blue one from here to here will be one segment. From here to here, next segment, and so on. Now, what they have discovered that engrailed, we know already, we learned engrailed is expressed in the anterior region of a parasegment. This will be engrailed in next parasegment, anterior of next parasegment, anterior of next parasegment. Now, what we have learned that engrailed, which is a transcription factor, it leads to activation of hedgehog, which is a diffusible signal, which then leads to activation of wingless. So the interplay between cells like this, wingless goes to the neighboring cell where engrailed is active, it activates hedgehog and hedgehog diffuses and activates wingless here. And here you have activation of hedgehog. Although the receptor patched, which is for hedgehog, is active here as well, is present here, but hedgehog signaling is not active here. It's only active in this cell where you have engrailed being expressed, because engrailed is the one which activates the hedgehog gene as well. So slightly after the parasegmental boundary has been established, the earliest sign of parasegment I told you are these tiny grooves in the embryo. The embryo looks normal, but there are slight grooves which are the earliest signs of parasegments. So this deep groove develops at the posterior edge of each engrailed expressing stripe. And that marks the edge of the segment, because this is the engrailed expressing posterior region. And that's where a groove will appear. A slight groove in the embryo will appear. And that marks the segment basically, because this is going to contribute to next segment. Now, this is what I was explaining you there. So if this is the posterior compartment of a segment, this is anterior compartment of the segment. So you will have engrailed, which activates hedgehog. Hedgehog diffuses. This line is basically the cell lineage restriction or compartmental boundary. Hedgehog diffuses and activates wingless. Wingless then diffuses and activates engrailed here, which activates hedgehog. If you disturb this signaling, like here, if this crosstalk or interplay between wingless hedgehog is not taking place, this is the phenotype. So this is the wild type. You see the anterior compartment of segment, and what we see that dentical belt is there, and then the posterior compartment is naked. Now, if you have mutation in wingless or hedgehog, you see a mirror image of this dentical belt, the mirror image. And this is next one. Already, what you are learning here is that the cells are not mixing beyond this compartmental boundary, which was defined here. All those cells are talking to each other. And these lineage restrictions or compartmental boundaries, they are the ones which remain intact in the adult organs as well through metamorphosis as well. I mean, you go from embryo to larva to pupa and then to adult, and you still have these compartmental boundaries intact. Isn't it is a miracle? So yeah, I told you already, wingless moves posteriorly over the compartmental boundary, as well as moves anteriorly to cells where it is expressed. So it moves there as well as there as well. Okay, but this crosstalk takes place only here. So wingless on this side is going to suppress the dentical belt formation, but here it induces the dentical belt. Similarly, if you look at the thorax region, let me show you thorax if we had fly. Yeah, here. Now, on thorax, you have these hair-like structures. But if you look at abdomen of adult fly, you will see pattern of hairs, which are very unique. On dorsal side, different, and then on ventral side as well. Very unique pattern. That is also due to the patterning or pattern formation within the compartmental boundaries. If you pay attention here, it's shown that one segment is based on these hair patterns. It's further divided into 6 different sections. For example, this one is A1. Next is A2, then these ones where you have bigger ones, A3, A4, A5, and A6, and this is the compartmental boundary. This is the engrailed expressing posterior, and this is anterior. You can generate genetic mosaics using flip FRT system, and you will see that you are going to disturb these hairs. They, instead of pointing all towards posterior, they will start pointing towards like this, a mirror image. Okay. So whenever I look at this particular figure, and the other one is polytene chromosomes, the way they have divided cytological positions of them, each band on polytene chromosomes, I can't help appreciating the intelligence or the genius Drosophilists who have spent hours on microscopes to define, these patterns and then defining where the compartmental boundary is. All this is tightly regulated at the level of gene expression. There's yet another example. Yeah, Friha?
Sir, I have a question. In the previous slide, there is this A1 to A6. What exactly are these? Like, I can understand totally that this is basically you mentioned that this is the thorax part and it's one of the segment, and then you have
No, this is not thorax part. This is abdominal.
So first you mentioned that we have bristles, the hair-like structures on the thorax part, right?
Mm-hmm.
So that structure is also present on the abdomen part.
Yeah, So the hairs on thorax are different than the hairs on the thorax. You will find 8 or so such big hairs.
Okay.
You can very each fly will contain same pattern on the
Okay.
thorax. In abdomen, there are more, and then these bristles. Not we don't call them hairs; these are bristles. These bristle-like hairs, they are much more on abdomen than on the thorax. The purpose
Okay.
of
So
bringing you
mhm.
this picture is to highlight that these compartmental boundaries are intact even all the way to adulthood.
Okay, and then yeah, another question. So these A1, A2, A3 to A6, can you please explain this as well? Like, this is not those segments that we have on the like
No,
the
no, no,
in the
no, no,
battles.
no. This
Yeah.
is basically one segment, abdominal
Yeah.
segment.
Yes.
These are further, they have morphologically, based on the pattern of these bristles, they have further subdivided each segment into just sections. We don't call
Oh,
them subsegment or whatever. This is just a
okay.
morphological classification or pattern, morphological pattern of
Okay.
these bristles in, let's say, segment one.
And every segment has a similar kind of a pattern, or it's a different pattern, as you
It's a
mentioned.
unique, it's a
It's unique.
unique, just like dentical beds, they are also unique.
Okay. Thank you.
So this is another example where you see this is the compartmental boundary. If the compartmental boundary is disturbed, you can see cells, they start mixing with each other. And this is the real picture under microscope, which is showing compartmental boundaries disturbed. And these compartmental boundaries are somehow the interpretation of morphogens which are expressed early on. They are the ones which establish such patterns. So with that, we are done with today's lecture. Uh, any question? Are there questions? GA Freya?
Sir, another question regarding you mentioned earlier in one of the slides that we can use a like we can use a fluorescent molecule and we can inject it in the single embryo, and then we can, locate where it goes during the developmental stages. So, but my question is like, like, are we supposed to like label that something with that fluorescent? Like we have to label something fluorescently and then, target it, or how does the fluorescent molecule,
So
move
these are
around?
called caged compounds. We get them ready-made, and
Okay.
they will definitely randomly go into few cells,
Okay.
and once you will use UV, that particular cell will be shining, or those few cells will be shining. You know now the precise location in the embryo where they are. And then as the cells divide and then contribute to different tissues and organs, you can trace them always with UV by activating them, and you will see, okay, the cell which was, let's say, in a specific region is contributing to this particular segment.
Okay. So basically, if you, let's say, because we have a syncytial blastoderm where there's just open, it's like a cytoplasm,
Yeah.
and if you inject at the interior side, so it had like a bicoid messenger, like the transcription factor, can it also like I don't know how to put it in. So can it also diffuse through the in a similar pattern? Like what are we actually using it for to study? Like exactly what?
So it's like you use these caged compounds to mark cells,
Okay.
to study their, presumptive cell fates.
Hmm.
So cells in posterior region are contributing to which tissues and organs.
Okay.
So
So
these compounds are just like analogous to in nowadays more sophisticated experiment we do is mark M clones. This is a genetic experiment to mark clones. Go and read what are Markham clones. I gave this assignment late in the later part of the course, but you can go on and read about this because you have to first do the assignment which I gave today, flip FRT clones. If you start understanding flip FRT, then you will know the Markham clones.
Okay.
Any other question? If there are no more questions, I wish you all a good day there.